So now we're in our new format again, and we're going to start over here, and we're going to work our way to over all the other places and stuff. So here we have early and late prophase. In early prophase, we're going to see our centrosome is going to replicate just before our prophase, and those are from early to late prophase. They're going to migrate from together to opposite poles of the cell. Now, also during this process, we're going to see that our chromosomes are going to be condensing. Chromosomes are going to start to appear, and they're going to be condensing. Our nucleoli are going to be disappearing. They're going to be present at the start of prophase, and they're going to slowly dissipate over the course of prophase. And as our prophase continues, our nuclear membrane is going to be breaking down, and our chromosomes are going to be separated. Our chromosomes are going to become very obviously a pair of sister chromatids connected at the centromere. All right, so that's what that prophase is going to look like. Now, if we switch over here, we'll see prophase with a look at the microtubule stain. So here we see asters forming. These are our asters. These are migrated centrosomes, and the centrioles are somewhere in there. And we'll see these strands of microtubules expanding outward. We also see the breakdown of the nuclear envelope and starting to see these dark spots right here, which is the condensing of the chromosomes. We can also see down here is a similar cell in a similar situation, breaking down of the nuclear membrane, etc., etc. So now we have an intermediate stage in prophase as well, where we have much more condensed chromosomes. We have fully migrated centrioles, excuse me, centrosomes, and we have these condensing chromosomes. Now, let's go back to our other cartoony side. Here at metaphase, as we discussed before, we still have our cell. We have completely broken down the nuclear membrane. We now have lined up our chromosomes, which are fully condensed, at the equator. They are lining up at the metaphase plate. And we have fibers. These are the kinetochore microtubule fibers, and these are the non-kinetochore microtubule fibers. The non-kinetochore microtubule fibers are connecting to the kinetochore. The kinetochore microtubule fibers are connecting to the kinetochore at the centromere. And the non-kinetochore fibers are not. That is the difference. All right? All of these are going to be migrating toward these poles over here. Now, let's look at metaphase over here in our different stain. So we have the asters still. We have lined up at the metaphase plate our different sister chromatid pairs of chromosomes. We have our kinetochore and non-kinetochore fibers, which we have a lot more fibers than we can legitimately look for and count. But this is our metaphase. We don't see any sort of plasma membrane whatsoever. We are, yeah, that's where we stand there. So let's go back to our anaphase over here. During anaphase, we have split the chromosomes from each other's centromere. They have been separated and they've been moved into a V shape. That V shape is because they're being dragged. If you tried to drag a piece of grass through water, then you would see that that grass bend and deform in shape as it gets dragged through the water. So that is what we're seeing as the kinetochore fiber pulls the chromosome that was formerly a sister chromatid by the centromere connected to the kinetochore to the opposite poles. These non-kinetochore fibers are still here in the middle, basically holding the other, saying, I'm not touching, no, you can't touch me. That sort of a deal. They're holding each other at bay. So now we'll move over to this anaphase over here. We see that these have separated and they are being pulled to opposite poles. That phrasing is very important. They are being, the chromosomes, the now single chromosomes, are being pulled toward opposite poles of the cell. Now, here we have another anaphase slide where they have been fully pulled to the opposite pole, we can see that they've scrunched with our centriole right there, our centrosome. So the non-kinetochore fibers are still there, but there are far fewer of them than we saw when we had our kinetochore and non-kinetochore fibers up here in metaphase. So note the difference between kinetochore plus non-kinetochore and only non-kinetochore. Now, at this point, we're going to move back to our cartoons, and we see we still have our centriole and our centrosome. We have our chromosomes are going to decondense at this point. That's not shown in our that's not shown here. And we're going to have the reformation of the nuclear envelope, and we are going to have the nucleoli are going to begin to reform. So we're going to see nucleoli again in telophase. So we're going to see that the nucleus has been the chromosomes have been separated completely, and they'll start to decondense enough that we'll start to see nucleoli reform. Now, cytokinesis is also shown in this image. Cytokinesis is a separate process that we'll be discussing in a moment. Now let's take a look at our stained image. So here we have cytokinesis is almost complete here. We have one set of chromosomes, We have the spindle pole and the spindle pole starting to decondense. The aster is no longer nearly as clear. And our chromosomes, are decondensing. I don't see the start of a nucleolus or a nuclear membrane forming here, so this would be I think that might be a result of the stain, just because it's really hard to see the nuclear envelope in this kind of staining anyway. We saw that in Prophase anyway, that the nuclear envelope wasn't particularly clear. All right, now let's see if there's, yeah. We'll come back to this format when we go back over plant cells.